Yeah, I think it will be less than that, because we discussed a lot this morning already about manufacturing, and I think most people are convinced that something has to be done for manufacturing and that this could be a good subject for the IMI. So the problem statement, manufacturing is still an obstacle to really make this ATMP a real world, I would say. Because, of course, there are not really the manufacturing means and protocols are not in place. And that means also that when you have to go to scalability, of course, that you have a problem. And scalability, although that it depends on if you go for viruses or you go for cell therapies, but then again, scalability is one of the big problems. And when you have the scalability problems, you also have problems with cost of production. So I guess that is one. Also, that regulatory is looking to it, although that, in my opinion at least, this is not completely developed. And so this is for me a field which is growing, and we should give it time to grow as well. But definitely manufacturing, quality, consistency concerns that there are. When do you decide that your cell is in the right differentiation state to be used in a patient? That's very important, I guess. So the scope, we are looking for the technological innovation in cell therapy and gene therapy, although that those are separate, but they have some things in common. Of course, you have to grow cells to produce your viruses as well. With that attention to closed systems, automation, and monitoring technologies. And during the discussion this morning, what we forgot a little bit is that there's a third party. So you have the pharmaceutical companies, and you have, of course, the others, but also you have the equipment providers. And when we had that discussion in October, we had some equipment providers around the table, and they are willing to invest in that as well. They are really willing to develop those closed systems. Because that's part of their business, that's part of what they do. So the emphasis is on therapeutic scale production and have the proper GMP standard in place. And to do this at a reasonable cost, of course, so that you also achieve your regulatory compliant. Which one I have to push? The green one? Okay. So these are some examples. So automation, and that came up during that discussion in October, is that there's a need in automation because automation means also that there's less variability in the outcome at the end. So that is something that is very important. And then you have, of course, two types of cells. You have the adherent cells, so cells that are in those cell factories, but also on microcarriers where you can grow the cells as well. But also cells in suspension. So that is looking for new processes in this. Besides that, robotized technologies, for some they are already in place, but not really for production means. So they are more in place in a lab when you start doing that. And also the large-scale banking of clinical-grade cell therapy is also very important, because sometimes you have to freeze the cells. And I know, and Wilfried can confirm this that so there was that project on chondrocytes, which was patient-specific, but you have to distribute your cells to the patients. So when the physician is asking you, I want to have the cells, you should transport them. So there's also something about good distribution practices. And also, and that's the difficulty, I guess, is we have a lot of cells that are used. We heard about the MSCs this morning, but you also have a lot of genes. You have a lot of gene therapies, which are different. You have only a few viruses. You have the most popular are, I guess, the lentivirus and the AAV viruses. But there, when you see how these are produced at this moment, I know from Unicure in Holland, which is an approved viral treatment, that they use two types of cells to really get to the amount they need to inject into patients. So that is something that they are looking at. But of course, you have that diversity, and one of the proposals was that, for instance, for the viruses, when you look at how viruses are purified, is that you could maybe have a kind of example virus which is used then, and where that new technology is developed to purify that virus, so that, for instance, if it's a lentivirus, that everyone who's using lentiviruses can use that type of technology as well. And then, of course, quality controls and standards, that you can use quality by design, do the proper risk analysis, and management, like the ICH is providing, and then you have the engagement between scientists and manufacturing. I have to say that we have a similar project coming up in call number 10, but that is not dedicated to the ATMPs. That is dedicated to the biomanufacturing of proteins. But also there, it's growing of cells, cell produce your protein, and that is where we start looking at how can we control this in a better way than we do right now to really have then a more consistent outcome, a higher quality product. So, but, and this could be linked to what is presented over here. But this is the program which is in part of the call number 10, is definitely dedicated to the CHO environment, so it's not to the ATMPs. So that's about it, I guess, if there are any questions, but I guess we have discussed this during the morning session a lot.
Thank you, Luc. Time for questions.
Luc, I have a question. On the deliverables, there is a lot of focus on the cell culture part of the technologies, the scaling up and so on. Would one estimate that this should also include here downstream development? I think for cells, the harvesting is one step, but especially purification of different type of vectors might also be addressed, or can we there learn from vaccine industry where technologies can easily be applied, and how is the thinking about that one?
Yeah, but that's included in this, but that's not included into what we do for the Biomanufacturing 2020 project. Yeah? That's for CHO, that's the upstream. But here we are looking at everything. So how you purify your viruses, because one of the things that happens, for instance, for viruses is that you have a lot of what you could call empty particles. So you can tackle this problem in two ways, I guess, by developing new cell lines, that could be part of it, but also by developing a better purification method to really separate the both of them. So those things are covered in. So these are just some examples, but it's much broader than that. And I think that once this project is becoming more real, that the partners should discuss what is needed. And how can we bring what can we learn from what already exists and the existing program? So, but it all has to do with how can we improve our manufacturing so that we have a more consistent product at the end. That is the goal, upstream and downstream included.
Mimo?
Yes, just to continue on the cell type and the producer cell lines, etc. I mean, one thing that I want to highlight is the issue of adherent cells and suspension. So most of the products for proof of concept are made mainly in adherent cells. The issue with that is that the many groups face is that when they want to move the product to the next step, adherent cells based production cannot be scaled up, and you cannot produce large quantities to take to phase 2, phase 3, etc. And they are forced to move to suspension, but again, that is a change in itself that will need regulatory consideration, etc. So maybe here we need to consider bring consistency in that as well, because that will allow to generate products that will be consistent and in the same, there will be less variability, etc. So suspension cells, maybe it's probably the way to go, but sometimes it's not straightforward.
Yeah, I know, but to answer, so the cell factories is one thing. You can also use microcarriers, and that's not really in the field of ATMP's use at this moment. We use it at a large scale. We have large bioreactors where we produce cells on microcarriers. So, and that's a 4,000 liter bioreactor. So that means it's feasible, but that depends a little bit. I think that when you transfer this from a cell factory to the microcarriers, then you can create problems. But if you can start earlier, we even have smaller bioreactors where we can use microcarriers as well. So we even at this moment use microcarriers at 5 milliliter scale. So in the AMBER system, I mean, and so it's feasible. I mean, but the only thing is that people are not aware that that is possible, I guess. And then if you go for suspension cells, that's the way to go. I mean, that is. But then you have, for instance, when you produce your viruses, you have to have how you do your transfection. How effective is your transfection? So that means that those are things that will be looked at in this as well. That's the idea. Yeah.
A question. When you say about huge diversity of cell and gene therapy products, do you mean a diversity of products or the heterogeneity of the population that give rise to the products, especially for the autologous one?
In the first place, the products, but also the heterogeneity in the products. If you take cells from a patient, how can you, so they are, per definition, there's a variability in there, per definition. So how this will react, that's also some of the things, and that is what I could call control, quality control of your cells. If you can, one way or the other, improve this and have a more singular population at the end, probably it's also more effective. So it's both.
Yeah, but there is also a question of the heterogeneity of, autologous patients. Each one is different. Each one gives different, well, the same product, but it's different. So how to address that kind of issue?
Everything what is from that is really a manufacturing problem, and that is what we try to solve with this project, which has not an agreement. But I mean, this again, it's a proposal, it's an idea. Other questions?
But this is one of the reasons that we don't need only personalized medicine with autological cells. We should also use the allogeneic side. There are different kind of effector cells which are going into the direction of the ATMPs. But what I wanted to say, I absolutely agree to this automatization of the manufacturing, but we all should be aware that we need then in such a consortium really the different companies, because if we discuss with regulatory what we, for example, just have done together with Milteney, because we brought CAR T cells to the prodigy, there are always also in these fully automated versions some bottlenecks which have to be solved as risk-adapted solution to really move forward. And this is the same if you go to the GMP upscaling for iPS cells with different companies which provide bioreactors like Eppendorf or so on. And so my personal opinion is that we really, if we go in that direction, we have to do it broad and to look what is available and to really sum up all these different experience to make a really good protocol for the different ATMPs.
Yeah, I think that's a good idea. I think that is where you can learn from. I mean, and for me, it's also a learning process. If you have a closed system, I think that sometimes regulators also have to look in how it works. And it's the same. If we look at how we produce monoclonal antibodies, it took a while to produce monoclonal antibodies at the scale they are doing now. And I mean, this needs some time to grow, of course, and also at the same time to convince that regulators start knowing what you're doing, and in that way that you together can bring that product more efficiently to the market. That is a little bit the idea, yeah, from a manufacturing point of view.